Very warm welcome to this, the second of our environmental microbiology lectures. Before we start, can I just say that if you happen to have one of these on you, could you possibly turn it off or turn it to silent, please? Thank you very much indeed. These lectures are co-sponsored by the Society for Applied Microbiology and also Wiley-Blackwell. They're a celebration of the achievement of the journal Environmental Microbiology, which, under the guidance of Ken Timmis and others, but primarily Ken, has become one of the leading journals in its field in a very, very short space of time. This year we are again delighted to welcome a truly outstanding scientist to present our lecture. Professor Edward DeLong is Distinguished Professor at MIT in Cambridge, Massachusetts in the United States. He obtained his bachelor's degree in bacteriology from the University of California and went on to do a PhD in marine biology at the Scripps Institute of Oceanography. Now, I thought I'd try and get a bit of background information on Ed. And I made a big mistake. I accessed his CV online and pressed the print button before I realized what a big document it was. In academic circles, scientific circles, the journals Nature and Science are usually regarded as the most prestigious journals, with all due respect to environmental microbiology. They are the ones that are generally regarded as the pinnacle of achievement. And in truth, most scientists would sell their grandmother for a paper in Nature. Now, to give you an idea of the stature of Ed DeLong as a scientist, I did a quick calculation and found that 1 in 5 of his published journals, and it is a mighty impressive list, were in one of those two journals. 20% are in either Nature or Science. Now, that is pretty impressive. In fact, I only went up to 2007, only because my printer ran out of paper at that point. The list of plenary lectures to which he's been asked to give talks at conferences ran to over 7 pages. The list of honours and awards he's received are equally impressive. They include election to the Fellowship of the American Academy of Microbiology, Fellowship of the American Academy of Arts and Science, and I understood from this evening that in 2008 he was elected to the National Academy of Sciences in the United States. This year he also received the David C. White Research and Mentoring Award from the American Society for Microbiology, which recognizes distinguished accomplishments in interdisciplinary research and training in microbiology. So Ed DeLong is an outstanding scientist who's radically altered the field of marine microbial ecology by bringing molecular biology approaches to the understanding of microbial communities in the oceans. And he's going to present the second environmental microbiology lecture entitled Deciphering Microbial Community Dynamics from Genomes to Biomes. Ed, you're very welcome.
Well, thank you very much, Jeff. That was quite an introduction, and it's really an honor to be here to give the lecture this evening. I do want to clear one thing up. I love my grandmother, and I would never sell her for any journal whatsoever, including environmental microbiology, Ken. So I want to thank the organizers and the Society for Applied Microbiology and environmental microbiology under the leadership of Ken for making this all possible in this very exciting time for microbiology. So thank you very much. What I'd like to do in starting out today, we'll see if we're wired up here, is give you a little background on why studying microorganisms in their natural habitats is important, lay a little context for that, and then kind of give you an idea of where we're at. What do we understand about microbes in their natural habitats? What things are missing? And in essence, where are some of the new technologies taking us for trying to answer some of these questions that have really been around a long time? Many of the questions that we ask in microbiology aren't necessarily new, but as has been the case historically in microbiology, really since the beginnings, is that Every time there's a technological innovation, and that could be pure culture technique, it could be an electron microscope, it could be molecular genetics, there has been a kind of quantum leap in our understanding of the microbial world, largely because we don't perceive the microbial world directly. We really have to observe it using indirect means. And because of that, some of these new technical innovations really help in our understanding of the natural microbial world. So to get started, let's back up a little bit and think about how we think about microbes relative to how other biologists think about the rest of the biological world that we can see directly. I think it's appropriate, now that I'm here in England, to quote a very famous population geneticist and natural philosopher, J.B.S. Haldane. And when he was asked what he might have gleaned about our Creator from his studies of science, allegedly his answer was he had an extraordinary fondness for beetles. And that is because of many of the species that we knew about in the insect world. There are about 10,000 different beetle species. So that's an interesting comment about species distributions in this invertebrate world. But let's think about something more close to home. How about us? Now Michelangelo didn't know this when he was painting the fresco, The Creation of Adam, on the Sistine Chapel. But the body of Adam and man is about 90% microbial cells and 10% human cells. And the only thing that one can infer from this is that the Creator had a truly inordinate fondness for microbes. Now, I'm not going to be saying a lot about human and associated microbial species, although what I will say, since I have this up right now, is that a lot of the techniques that I'm going to be telling you about have been applied to the ocean, because that is the environment and the microbial communities that my own lab particularly studies. But many of the techniques I'm going to be talking about have very particular and applied applications to understand, for instance, the relationship between us and our microbial species, and how, in fact, they may be involved in particular diseases. And most recently, Jeffrey Gordon's group has been showing the relationship between shifts in microbial populations and how that changes how we process energy and its relationship to obesity. So many of the techniques I'm going to be talking about have very applied applications, more so than what I'll be talking about in the ocean. But I would argue that what we're trying to understand in the ocean really is, on a global scale, the energy and matter fluxes that are occurring that have to do with global biogeochemical cycles. And of course, since the dawn of the industrial age, mankind as a species is now pushing as hard in terms of mass and energy flux on biogeochemical cycles as are microbial species. So understanding the rates and fates and the organisms involved in these processes has been there hasn't been a more critical time, because we're pushing on these cycles just as hard ourselves now. I may get to that point in a bit, but natural cycles that we know about in terms of fluxes of energy and matter started on Earth pretty early. We think the Earth's about 4.5 billion years old, and the evidence suggests that life probably arose somewhere around or before 3.5 to 3.8 billion years ago on Earth. So for most of Earth's history, life has been around. And in fact, many central life processes like photosynthesis evolved very early in the evolution of the Earth system. The Earth system being the geological component and the biological component interacting over time. That's been going on for a long time, and it was by probably maybe 2.8 billion years ago that evidence for oxygenic photosynthesis can start to be seen in the fossil record and in isotopic records. So that the activities of these very small microbes that are distributed on global scales, they kind of act in a massively parallel sort of way to regulate flux of energy and matter. And of course, the evolution of cyanobacteria are what led to the 20% oxygen in our atmosphere and allow us to breathe, basically. So the microbial world and its evolution has had a tremendous impact on the state of the world that we see around us today, partly because of this long evolution. Now, when you count microbial numbers, they're vast and almost unimaginable. There are more, for instance, microbes in the ocean than there are stars than we can see in the sky, somewhere around 10 to the 29th or so cells. And these are not just static entities. In surface waters of the ocean, microbes turn over on the order of about a day. That is, they have a growth rate of about a day, and they get consumed by small flagellates or ciliates at the same rate. So the turnover of cells, particularly in surface waters of the ocean, is incredible, somewhere around 7 times 10 to the 29th. A million moles of cells turning over. And if you think about the dynamic processes that are going on as these cells are turning over with respect to mutational events, lateral gene transfer, the invention of new genes and pathways, there's a tremendous amount of grist for the mill here in terms of evolutionary process, much of which we don't fully understand yet. So there's a lot to learn. And of course, now we can look at the Earth from space, and what we see basically are vast areas like this. And what you could see is the ebb and flow of this chlorophyll that's compressed over time, over about 3 years or so. And you see basically the upwelling of nutrients that causes phytoplankton blooms, leading to huge amounts of biomass that, of course, feeds the rest of the food chain. And really the point of showing this slide is that we can look from space and watch microbial activities that feed the rest of the planet. And by the way, provide these organisms in the ocean provide about every other breath that you take with respect to oxygen. Maybe 50% of the oxygen is produced in the ocean, the other 50% by terrestrial plants. So these are tremendously important processes. And so the question is, well, how much do we understand of what's going on, what particular organisms are there, microorganisms in the environment, and which processes are they involved in? And one kind of conundrum that's been known about for a long time in microbiology is that traditional plate counts and cultivation techniques often don't recover all the organisms that are in any particular sample. In the case of the marine environment, if you take a milliliter of seawater, you might get on the order of hundreds of colony-forming units per milliliter. If you take that same milliliter of seawater and use DNA staining dyes that fluoresce when they bind DNA and count the number of cells, you get about 1 million cells per mil. So we're off by many orders of magnitude in terms of what we can grow as colony-forming units on standard media compared to what is actually there and what we can count. So the question for quite some time has been, what are we missing? And a lot of the technologies that I'll talk about really are getting at that in terms of understanding who's there, what they're doing, and how that relates to ecosystem processes. And that ecosystem could be the ocean, or that ecosystem could be the human gut. Well, in the mid-'80s or so, molecular phylogenetics became very well developed so that we could basically start to compare all cellular organisms on one tree of life. And that was by looking at macromolecules that are highly conserved and were present in the ancestor of all cellular organisms on Earth. And many of these molecules are known as ribosomal RNAs, which are present in ribosomes, the same ribosomes that were present in the ancestor of cellular forms of life on Earth that we know about. So one can sequence those ribosomal RNAs and get a metric, basically, of evolutionary distance. And since all cellular organisms have ribosomes, you can put them all on the same map to look at their relationships. So we can look at a quantitative evolutionary distance between, say, E. coli and man down here. And what this has allowed microbiologists to do is to put microbes in an evolutionary framework and relate them to one another and to larger organisms as well. But what Norm Pace realized in the mid-'80s or so was that you could use this molecular phylogenetic approach to identify microbes even if you couldn't grow them. And the scheme is quite simple, and this is actually from a paper of Dave Stahl's in 1985. And the scheme is that basically you can reach into a mixed natural population, collect that biomass, extract total DNA or total RNA, and let's not worry about the details of the methods here, but the bottom line is you can extract from that DNA these phylogenetically informative macromolecules that tell you who's there, what kind of organisms are there. You can sequence those molecules and put them on a tree like this to say this sort of organism and that sort of organism was present in this mixed natural population. And so for the first time, we could take a fuller inventory of indigenous microbial species without being limited to whether they would grow or not. So we got a fuller picture of biodiversity of microbes in the environment. And that has had a very profound impact on what we understand about microbes today. Back in 1987, before those techniques were applied, we knew of about 11 or 12 major bacterial phyla. These would be things like the gram-positive bacteria, the proteobacteria, cyanobacteria, Cytophaga flavobacteries, and so on. 12 major phyla in bacteria, or major divisions. As people went out and applied these cultivation-independent techniques, this is how the extent of our knowledge of major evolutionary lineages has changed over time. And what you see here is a tree, this is somewhat dated right now, but up to 2006, there was something on the order of 60 to 80 major phyla. These red here represent those which haven't even been cultivated yet. So we don't even have representatives to tell us about the biochemistry, the physiology, the ecology, and the genetics of these particular major lineages. And of course, within these lineages are many orders, family, genera, species, and so on. And what I like to tell my students when we ponder this fact is that their students will still have plenty to understand and explore about the microbial world, because this is a rather humbling realization in terms of what we truly understand about extant microbes on Earth. Given that problem, that we only have snippets of ribosomal RNAs that we can put these organisms on a phylogenetic tree, but we really don't understand much about their biology, many scientists have been interested in trying to advance methods that will teach us something about the biology of these unknown groups, many of which, by the way, are major representatives in different habitats. So these are not trivial species or rare species. Many of these are quite dominant in particular habitats. And one approach that people have used are applying the techniques of genome sequencing to looking at mixed natural populations. And this is basically the same approach of Pace et al. For looking at ribosomal RNA genes, except trying to extend it to the whole genome. So one starts with an environmental sample of a mixed natural population, extracts DNA, sorry about that. There's a variety of different techniques for sequencing that one can use now. And from these mixed bag of sequences that you get from the environment, there are several approaches one can take. You can try and assemble those reads into an entire genome. That's an extraordinarily difficult thing to do for many different technical reasons, which I won't get into, but this has been done. One can simply take a kind of a gene-centric approach and ask, well, in this particular environment, what panoply of genes do we see that tell us something about environmental interactions and types of microorganisms? So you can just focus on individual genes. You can also bin particular fragments to look at particular lineages, even though you can't assemble them fully. You can look at groups of similar clusters of fragments, genetic fragments, to try and understand who the dominant players are in a particular environment. So this sort of approach actually has opened up new windows for understanding what sorts of things are happening out in the natural environment. And I'm going to tell you actually a pretty old story now, but it's illustrative of what we can learn about microbes in natural habitats that we really didn't fully appreciate before. And in this particular approach, we were working, I was then working at the Monterey Bay Aquarium Research Institute, and we were working at several sites out in the bay to understand the sorts of organisms that were there, microorganisms, their natural fluctuations, and how that might relate to processes ongoing out in the bay. And so we took this approach of collecting the plankton, the microbial plankton, between about 2 and 0.2 microns. That contains most of the bacteria and archaea in these samples. We would extract the DNA, but we would try and get very large genome fragments on the order of maybe 5 to 10% of a whole genome. and clone those into what are called bacterial artificial chromosome vectors. These just allow you to archive very large pieces of DNA representing significant fractions of microbial genomes present in the original population. And what we were happy to see was that when we surveyed these back libraries for ribosomal RNA genes, we found these very large genome fragments contained ribosomal RNA genes that were from groups that we knew were present based on polymerase chain reaction surveys. So these were groups that we knew were out there. They hadn't been cultivated, but the only information we had were ribosomal RNA genes. Now these are shown in these black boxes, so we got very large genomic DNA clones from all these different, at that point in time, uncultivated groups. And that allowed us access then to the genome content to ask the question, well, does the gene content of some of these groups that we haven't been able to culture yet tell us something about their physiology, ecology, biochemistry, and so on? And in fact, this particular group right here, it's called SAR86. That just refers to where it was first observed in the Sargasso Sea. This is a proteobacteria, fairly closely related to Vibrio and Oceanospirillum, same group that E. coli is in. This organism is found throughout the world's oceans, mostly in the surface water. Still hasn't been cultivated yet. But what we were able to do was take a genome fragment that was about 140 kilobases, maybe 5 to 10% of the genome, and sequence that. And when we did that, we could walk from the ribosomal RNA, and this, of course, told us that this is a SAR86 organism based on phylogeny, and look at gene content. And many of the genes that we found were not so surprising. They were typical functional genes, things like ribosomal proteins or respiratory genes that are also found in other gamma proteobacteria. But what was surprising was the presence of a rhodopsin in this bacterial genome fragment. Now, this was about in the year 2000, and it was surprising then because no rhodopsin had been found in any bacteria up to this point. So that was kind of surprise number one. But what intrigued us most was that what we understand about microbial rhodopsins from archaea suggested a potential functional role for this molecule, this protein, in these organisms that might relate to their ecophysiology. Let me explain to you what I mean by that in a second. The other rhodopsins that we knew about were actually in microbial species, prokaryotic species, came from halophiles, extreme halophiles that belong to the archaea. And you can often see these in salterns and evaporite ponds. When the salinity gets high enough, these organisms will bloom, these haloarchaea. And although much of this color comes from carotenoids, some of this color comes from these rhodopsins, these photoprotein pigments that are found in haloarchaea. And these are really, really interesting proteins because they're probably the simplest photosystem that we know about, a way for an organism to garner energy from light. And the way that these rhodopsins work in haloarchaea, bacterial rhodopsins that is that they're 7-pass membrane proteins, and they're bound to a chromophore retinal, the same chromophore we have in our eyes, actually. It's a vitamin A derivative. This retinal, covalently attached to the protein, when a photon hits it, undergoes an isomerization, and that isomerization causes protein conformational changes that ultimately result in the transport of a proton from the interior of the cell to the exterior of the cell. So a single photon of light can result in pumping a proton outside the cell. The reason that's important is that the way that all cells generate energy, including the mitochondria within our own cells, is by generating a proton motive force. Now, that proton motive force can be used, for instance, to synthesize ATP, to transport amino acids, to drive flagella. So here you have a single protein with this one retinal chromophore that can take light and turn it into cellular energy. This is now in the haloarchaea I'm describing. But when we found a rhodopsin in the bacteria in surface waters of the ocean, our first thought was, well, does this have something to do with the energy physiology of these cells? Well, if you put this proteorhodopsin, as what we dubbed this bacterial rhodopsin from these marine bacteria, onto a phylogenetic tree, you really couldn't tell how it might function, because it turns out that there are several different classes of rhodopsins in the haloarchaea. One is that one I just told you about, the proton pump. There's also chloride pumps, as well as sensory rhodopsins that allow haloarchaea to phototax. So we couldn't tell just from the primary sequence or looking at trees, phylogenetic trees, what the function of this new bacterial rhodopsin might be. But we got pretty lucky because Oded Beja in the lab, he's now at the Technion Institute in Israel. He was a postdoc, and he was able to express that rhodopsin in E. coli. When he did that and added retinal, what we saw is this typical chromic shift that you see in these retinylidine proteins. That's when the retinal binds to the protein. You see a dramatic shift in absorbance at about 520 nanometers. And this just shows that this protein that we got from this marine bacteria in the ocean was functional with respect to binding the chromophore. But what we could do was simply copy some of the early classical experiments that were done with haloarchaea. And so when Oded added that proteorhodopsin to E. coli, expressed it in E. coli, added the retinal, and turned the lights on, we could see proton pumping over very short periods of time, which suggested that this protein was indeed a proton pump and could generate a proton motive force simply by absorption of photons, that conformational shift I told you about, and proton pumping outside the cell. I won't get into the details here, but we had to convince some biophysicists that this was indeed a pump and not a sensory opsin, and you can do that by looking at the kinetics of the photocycle. I don't want to get into the details now, but the bottom line is that the answer is that this was a pump very comparable in terms of its kinetics of the photocycle to bacteriorhodopsin, and that our model then became that proteorhodopsin, this marine bacterial opsin that we found just using genomics, was indeed a proton pump and could fuel cellular energy. And I'll elaborate on this a little bit more. The point I want to make here, though, is that with these new genomic techniques, it really, you make an observation of a particular protein based on homology, and that in itself can lead to hypotheses, but that's all they are until you test them. And we were quite lucky in the sense that we were able to test the hypothesis that this was a proton pump, and it was able to be expressed quite readily in E. coli, but we were able to take it through a little bit more biochemistry and physiology to back up what we saw from merely looking at sequence data. Once you know about things in the environment, you can start to ask really interesting questions. So it turns out opsins, like the ones we have in our eyes, are spectrally tunable. That is, they can, you can change just a few amino acids and get very large shifts in absorption spectra. And in fact, that's one of the reasons why we can see color so well. And if you look at the opsins that we found out in the central North Pacific Ocean, when we sampled at the surface, we got the predominant rhodopsins that we found absorbed out here in that green, like I just showed you. So the absorption maxima is around 520. But when we looked at the rhodopsins that we could basically amplify and clone from deeper in the water column, they were spectrally shifted such that they absorbed around 490 nanometers or so. And in fact, that perfectly matches the absorption, the spectral irradiance of light at that depth. So light, as it penetrates the water column, gets attenuated, and you get basically the penetration of the shorter wavelengths at depth. And the opsins, it looks like, have been selected and tuned to absorb that particular light at that depth. So once we realize that there's a particular functionality, we can start to look at its variation in the natural environment and where it occurs in different microorganisms. After we made these observations, Craig Venter went out on his legendary expeditions, first in the Sargasso Sea, and in the very first samples they looked at, they found a huge variety of these very same opsins that we found. Now, we were looking in a very targeted way, and so these were the initial opsins that we found that I just told you about. But there are many others out there, too, that were found in the course of the Sargasso Sea survey. So the genomic kind of picture is opening up this world of photoproteins and what their functions might be in these marine bacteria. I don't want to get into all the details, but suffice it to say, with access to this material and the ability to express it in E. coli, there's all sorts of things one can learn. And Oded has gone on to do site-directed mutagenesis, and it turns out you can change one amino acid, this leucine, to a glutamine, and get the same spectral shift that I just showed you from about 490 to 520 nanometers. And although we can do this in the lab, nature's been doing this for a long time. So here's a tree of amino acid sequences of rhodopsins from the Red Sea and from the Pacific, and you can see the variance in spectral absorption, even though these molecules are almost identical in sequence. Because of this one amino acid shift, you get huge changes in absorption. And by studying these molecules in different species or in vitro, we're starting to learn quite a bit about whether or not they're sensory, and it appears that most of the marine rhodopsins are not sensory based on a variety of evidence that I won't get into. And we're also learning that they don't travel alone. So in about 30% of the opsins that we've studied so far using these genomic clones, the genes for biosynthesis of the chromophore are linked. So retinal is synthesized from basically beta-carotene. And so in many and the other thing I should say is that these opsins are distributed across many different species. So planktomycetes, beta proteobacteria, alpha proteobacteria, even archaea that live in surface waters of the ocean appear to have acquired these genes. And in fact, they've acquired the whole operon, which requires about 6 genes to basically biosynthesize the whole photosystem unit. So you have these genes here for synthesis of beta-carotene, which makes the chromophore. This gene here cleaves the beta-carotene to retinal, and then you have the proteorhodopsin here. So it doesn't take much, it turns out, theoretically, to shift from being a heterotroph to a photoheterotroph. In fact, all it, in theory, takes is this set of 6 genes or so. And it looks like it's happened in many different species. And in fact, we have evidence that these genes are being laterally transferred based on phylogenies of each of these individual proteins. So these genes are moving around, and they're getting selected, presumably because of the fitness advantage one gets when you can make energy, biochemical energy from light. And in fact, we've shown that we can demonstrate E. coli can synthesize ATP from light by transferring this suite of genes into it under starvation conditions in order to pick up the signal. So it doesn't take much to really switch, it turns out, to being able to use biochemical energy from light. And in fact, that's happening, we think, in marine surface waters all the time, with different species acquiring this particular capability. And the sobering thought is we didn't even know of the existence of these particular genes in bacteria or this process in the marine environment until just a few years ago. And it really does alter the way we think about energy transfer in these particular systems. I'm not going to go through this, but since a whole bunch of full genomes have been sequenced, what we're finding is that many different sorts of bacteria, even very typical ones that we grow in the lab in plates like Vibrios, also contain these very same photosystems that I just told you about. So we have a lot to learn. And part of what we're learning comes from this combination of techniques and approaches and philosophies, really, that have been around for a long time. Mapping genes onto organisms started with Mendel, if not earlier. That's simply genetics or genomics. Mapping organisms onto the communities or environments they live in is really ecology, and that's been going on for a while too. And what's a little new that I just described to you is this business of mapping genes onto communities in the environment, commonly referred to as metagenomics now. And the sweet spot is where we can combine all these sorts of different information to understand the relationship between the genes in the organisms and the environment, and how those all work together to give the patterns that we see today of organismal distributions and gene distributions. Okay, so that's kind of a gene-centric approach, just to convince you that one can learn something from these sorts of approaches and technologies. I want to very briefly describe to you work that's been going on with my colleague Dave Carl north of Hawaii in the Central Pacific. This is a patch of the ocean that's representative of vast areas that cover our planet and are very important with respect to energy and matter cycling. And they're dominated by microbial species. So the microbes dominate biomass in many of these areas. And what we wanted to do was simply walk a kind of environmental gradient, if you will, to see what sort of signal we might pick up from the genes that we could collect from the mixed microbial communities. And so we took samples from a variety of depths up in the upper water column here, as well as in the lower water column. And I'm just going to very briefly, this is a bit messy, and so we don't want to get lost in the detail, but what we're looking at here are the collection of genes that came from either 10 meters, 70 meters, 130, and so on. And then these are categorizations, and I know that you can't read this, don't worry about it, but basically think of each of these lines as a functional category of genes. So we're trying to compare functional categories of genes obtained from these different depths. And what I want to show you here in these blow-ups is that we do see different patterns and that they tell us different things. They can tell us about the sorts of organisms that happen to be at these different depths, or they can tell us about processes that are prevalent in particular environments. And in the case of organisms, there's this very large signal here, it turns out, of this transaldolase. And that wouldn't mean anything right off the bat. It's a transaldolase that's involved in basically carbon backbone configuration in terms of the pentose phosphate cycle. But this particular transaldolase, it turns out, comes from marine cyanobacteria, in particular Prochlorococcus. So if you dig into this signal at 70 meters here, what we're really looking at is the signal of cyanophages. That is, these viruses that are infecting Prochlorococcus at this particular depth. And I won't go through the data, but what I can tell you is that we happen to sample at that particular depth when there appeared to be kind of a massive Prochlorococcus viral burst happening. I won't show you those data, but this is just to indicate that some of the signals that we see told us about the sorts of organisms that were there. In this case, this was a signal from the phage that infects cyanobacteria. When we looked at other patterns, we saw other kind of distributions that told us about how genes might vary as a function of habitat across whole communities, which is a different way to think about things. And in particular, what we saw was there was almost a monatomic increase in terms of one particular gene category, in this case, transposases. Transposases are jumping genes. These are mobile genetic elements, sometimes called selfish genetic elements. And this is a curious observation because what it suggests is that within the genomes of organisms, as you go greater and greater in depth in the water column, you're going to find more and more transposases. And we've looked at these data sets and parallel data sets very carefully, and this seems to be a real signal. And it's a signal that's not pointing to any particular taxa. So that if you look at many of the different taxa that occur at these depths, what you find is greater numbers of transposases within their genomes as you go deeper and deeper. And also there's many families, so it's not just one kind of transposase, but something like 70 or 80 different families of transposases that seem to pop up. Now, why is this interesting? Well, to us it suggests this particular gene signal that it's telling us something about the selective pressures. In this case, we think a relaxed selective pressure against transposases. So that for whatever reason, and it turns out as you go deeper, population densities are less, so the effective population size of microbes is less, and also these organisms are more energy limited. Under those conditions, you see increased numbers of these mobile genetic elements. And that's something that one can follow up further because there are parallels, it turns out, in the world of symbioses that reflect kind of similar patterns. Again, I don't have a lot of time to talk about it, but it just shows you that if you walk an environmental gradient, you can find patterns of gene distributions that cut across the community and tell you more about selective pressures potentially across the whole community. This speaks more to the phage that I told you about. I told you about that transaldolase. And so, in fact, in these particular genetic libraries, in these genome fragments, upwards of 10% of them at 70 meters actually originate from cyanophage, it turns out. And that's this black line here. So we hit some kind of a viral burst, we think, because this was a cell fraction. We weren't even collecting phage, so we were collecting on a 0.2 micron filter, and what we think is that the Prochlorococcus we collected here were chock full of their viral parasites, basically, at this point in space and time. But again, one can pick up signals like this to look at even host-parasite interactions. I want to finish up in terms of some of the approaches that one can use. Now, I've been telling you about the approach of looking at the genome, basically, at the DNA. But of course, there are other levels of information flow within cells that give you finer granular detail and different sorts of information. And of course, DNA gets transcribed into RNA, which eventually is the message that turns into protein. And with new technologies available, one can access this RNA fairly readily. And what this can tell you is, at any given point in space and time, which of these genes within the DNA is actually being turned on or being expressed to synthesize its cognate protein. And what we think is that the information flow that you see, just in terms of the DNA that's there, but also in terms of what's on or off, tells you something about the particular biotic niche space that these organisms live in. And of course, other groups, Jill Banfield's most notably, are using proteomics, that is actually extracting protein from the mixed population to learn more about which proteins are expressed as well. And of course, each of these different layers of technologies has its own particular problems, difficulties, and challenges. But I'm going to talk about this one here, that is trying to look within natural microbial communities about which genes are being expressed. And this in some ways is pretty exciting, because if you think about microbes as potential canaries in the coal mine in terms of environmental process or insult, then this is a very sensitive way to try and tap into that. So the approach is fairly simple. What we typically do is take the mixed natural population, extract community DNA, and use some of these genomic techniques I told you about. One can at the same time extract community RNA, and because of some new advances in kind of methodological procedures, you can take very small amounts of RNA and get enough material to go ahead and sequence. The reason why that's important is you want to try and minimize perturbation of the community while you sample. And so we've pushed very hard to get to the point where we can use about 100 mils to 500 mils of seawater, which we can filter in just a couple minutes to do these analyses. And of course, we'd like to do it faster. You're always worrying about how you're perturbing a sample or a community when you're sampling, but there are practical limitations. So we can take on the order of a couple hundred mils of seawater. That translates into about 10 to the 7th cells or so to get information back out. And when you do that, one thing that you can do is take the number of messenger RNAs now converted to cDNAs that you see, and divide that by the same DNAs that you see in the same sample to get a ratio of expressed proteins. And basically, we map these onto well-known protein families that have been found in the marine environment, because that's where we get the most hits, basically. And if you do that, you can take a look at more highly abundant transcripts, those that are kind of in a medium range, and those transcripts which are at lower abundance, and you start to see patterns, some of which make sense. That proteorhodopsin I told you about, it turns out in surface waters of the marine environment, and not deep, are highly expressed, which makes sense in terms of their interaction with light that we know about. Many of the genes for carbon fixation, electron transport in photosystems, and so on, are also highly expressed. So this is just in survey mode to see if this technique really works. What you can do as well is take a whole genome and map those transcripts against the genome. What that tells you is, well, what kind of coverage am I getting? Do I see most of the genes within a genome being expressed or not? And what does that tell me about what's going on? And the bottom line with very abundant organisms, like these marine cyanobacteria, much of the genome is being expressed, and at very different levels. And in fact, if you take a look at which genes are being expressed when, and I'm not going to show you the data, this is in collaboration with Penny Chisholm, you can actually tell what time of day it is. It turns out these Prochlorococcus are on a diel cycle. There's a circadian rhythm. And you can map which genes are being expressed at which points in time in laboratory cultures. And it is like clockwork. And if you go out to the field and you take a sample, you can do a correlation analysis and basically pretty closely nail what time of day that sample was collected. And when a graduate student, Maureen Coleman, first showed me this, I told her that was an expensive way to tell time. But it's quite remarkable, and the point here is that we can ground truth some of these approaches in the lab and then go back to the field to see if things are really working or not. And this has really been quite remarkable. And so we've looked at a variety of depths at different sorts of transcripts, and you can get similar sorts of curves, and we see at different depths different genes being expressed, not surprising. And they tell us about what's going on in the environment. I don't want to walk through this, but these three samples that represent a transect through the photic zone with different kind of abundances of transcripts coming from different organisms. When you get down to about 500 meters, the situation changes dramatically. And in fact, ammonia oxidizing archaea represent some of the most abundant transcripts that we can find. So ammonia metabolism, nitrogen metabolism, heterotrophy starts to raise in terms of the signals that we see relative to what one sees in the photic zone, not surprisingly. I think I'll finish up by saying a little bit more about how exploratory this all is, because when we started to look at these data sets of messenger RNAs that we could sequence from the environment, we started to find some holes. And so in these RNA data sets, a good fraction is ribosomal RNA. Another pretty good sizable fraction are protein coding transcripts, messenger RNAs, if you will. But then there was quite a number of unassigned reads which we didn't understand at all, and we didn't know what they were. And that got us quite worried because there's lots of room for artifact in these new sorts of approaches. And so what do these unknown RNAs represent? Well, what we started to do was look for patterns. And when we took those unknown RNAs and mapped them against scaffolds of DNA sequences, what we saw is they piled up all to one particular different particular regions in different genome fragments, but all in the same place. And those same places, not surprisingly, were in intergenic regions. So remember that these fragments didn't hit anything in the protein databases. They didn't appear to represent proteins. And in fact, when you find a scaffold that they match in a DNA sequence, they fall between the genes. So these are RNAs that are being expressed in areas where there is no protein coding region. And they represent things like these small RNAs. This is a catalytic RNA, RNase P. Some upstream regions that you find in tRNA synthetases and other genes. Other small RNAs that have been found in things like Prochlorococcus. So what is this business with small RNAs? Well, it's a new area that people are exploring quite a lot now in the microbial genome, because the small RNAs represent a different way to regulate gene expression, in essence. And so there are entities called bacterial riboswitches, for instance, which are small upstream regions of RNA that can fold, and they actually can bind substrates. And when they bind substrates, they alter conformation and can turn on or turn off gene expression. So this is a really different model than what we're used to with respect to protein regulators of gene expression, because these riboswitches can actually are just upstream RNA sequences that regulate the turning on and off of particular gene suites. And so when we looked, and I'm sorry, this table's not going to be very palatable, but just suffice it to say that when we looked in these transcripts that didn't map to proteins or ribosomal RNA, many of them did indeed map to known riboswitches, for instance. Glycine metabolism riboswitches, other sorts of entities that are in databases of these small RNAs. So that was neat. That verified that some small percentage of these RNAs that we found were actually known small regulatory RNAs, but only a small percentage. So maybe 1% or so here, 2% or so here in this green wedge of these unassigned reads actually match these known small RNAs. But what we then wanted to do was figure out what about the rest of these RNAs. And again, that table's not so good. This one's not much better. This is hard data to present, actually. But the bottom line is we could take those unknown sequences and cluster them together by similarity, and we get very large groups of over 100 sequences. And you can look at those groups and identify features that are known to be present in small RNAs. And one of those is compensatory base changes that tell you about how the molecules fold up on one another. And some of those groups of very similar small RNAs were found across all depths that we looked at. Some were found to be depth specific, so on and so forth. So we could begin to characterize these small RNAs with respect to where they are, what organisms they're associated with, and what functions they might be involved with. So now we can create these maps from these transcriptomic data sets that tell us not only which genes in a particular genome, in this case this is Pelagibacter, a very abundant heterotrophic bacteria in the marine environment. We can tell not only which genes are being expressed, but also which small regulatory RNAs are abundant at any particular point in space and time that we're sampling. So it kind of opens up a new world in terms of regulation of RNAs in these natural microbial populations that, again, might provide us clues about how genes are being turned on and off, but also what environmental conditions are conducive to the sorts of patterns that we see. So I think what I'm going to do is wrap up very briefly. I did have a bit to talk about in terms of carbon cycling, but I don't want to run too far over. But I will talk about the approach that we're using because it's really leading to some fun stuff. And this is taking all the sequencing technology and not doing surveys like I just told you about, but actually doing experiments so that one can set up a control and a treatment. One can track what the community does over time by sequencing to learn about the dynamics of which organisms come up and how that relates to which genes are being expressed. And when you get patterns from a data set like that, it immediately suggests what you need to do for the next experiment. And so I guess what I'm saying here is that a lot of this omic technology, genomics and transcriptomics, has been in survey mode in terms of sampling. And now we're kind of entering into more experimental mode. And I'm not going to talk a lot about it, but this is briefly the approach where one can take a microcosm, basically, a control and a treatment, maybe 20 liters of seawater, for instance, incubate it under the right light and temperature conditions that mimic the environment, and take samples of RNA and DNA and look at the different expression patterns to see what they tell you. And the bottom line, without going through all the details, which I won't, I'll spare you, is that we've been able to figure out a big pool of carbon in the ocean, it turns out, is dissolved organic carbon. And that represents as much carbon on our planet as CO2 in the atmosphere. So it's a very large pool of carbon. But we don't know much about which organisms are chewing it up and what biochemical pathways they're using to do that. And to make a long story short, we've been able to try and ferret out which organisms are actually doing it. It turns out it's a succession of organisms that are involved in the breakdown of dissolved organic matter within communities. And one of the end pathways, it turns out, is methylotrophy, so that some of these dissolved organic matter compounds are made of methylated sugars, which appear to release carbon compounds like methanol. And at the end of an experiment like this, we see all the genes for both dissimilatory and assimilatory single carbon compound metabolism coming up in a very concerted fashion. So you get a peek into the organisms as well as the pathways that then suggest future experiments to try and advance our knowledge of really the ecophysiology of microbial communities. And that's really important because it's the ecophysiology of microbial communities that drives much of what we see in terms of energy and matter cycling in the ocean, or for that matter, energy and matter cycling within our own guts potentially. So I think what I'll do is leave it there and say that, you know, I've been focusing on these one set of approaches, and it's a bit myopic. There is no one technique that fits all and that will answer all the questions that we have. But these particular techniques are very generic in the sense that all organisms have DNA and RNA, and we can access those more or less equally with some caveats. Because of that, the information encoded in that DNA and RNA is accessible and allows us to go from just looking at these genomes or transcriptomes to making inferences about community metabolism, about organismal interactions, and about how these all combine to drive processes in any particular habitat or environment one is interested in. So it's exciting times to be a microbiologist, and we all have a lot to learn. And it's an important time to do that because while microbes for about 3 billion years, 3.6 billion years have been driving all these cycles of energy and matter on Earth, they've got heavy competitor now. And we don't understand how that all works still, and time's running short. So these are all the folks that did a lot of the work that I talked about. I don't have enough time to go through names now, but I'm incredibly lucky to be working with such a fun group of energetic and talented people. And I'm also very lucky to have enough funding to do that and support them. And so I'm very grateful to my funding sources as well as to the folks that have done all this work. And with that, I think you're probably all ready for me to be quiet. But I'd be happy to take questions. Thank you.